Okay, so our next speaker is Grace Wu, who is a trainee in Heiyuni's lab at the University of Toronto, and she'll be speaking to us today about the extracellular domain of the GP1B alpha receptor. Grace?
Good afternoon, everyone. First, I would like to thank the symposium organizer inviting me for giving me this opportunity to give a talk about our lab's recent work published in PNAS. It's actually the first time I joined this symposium. Actually, I learned a lot from previous speakers, and there's a lot of things going on. Like for the research, it's really exciting because actually I graduated from a PhD degree of immunology, so in this field it's still kind of new to me. So it's really a great opportunity, like for me to talk with you and to learn a lot of things from the excellent scientists here. So today, hopefully at the end of the talk, I can convince you that the GP1B alpha extracellular domain and Kupffer cells are interdependently necessary for platelet-mediated TPO generation. So for this audience, we all know that the TPO is a key hormone to regulate platelet production and is required for megakaryocyte differentiation and also the long-term maintenance of the hemopoietic stem cell niche. And also we know that this hormone is tightly regulated and is fine-tuned through the platelets as well as the megakaryocyte internalization. And also in recent research, we'll also figure out, so platelets not only can internalize the TPO, it can also induce the TPO generation from the liver. So in Dr. Ni's group, the previous PhD student Miao Xu, and published a paper in Blood in 2018, and her research pinpoint that the GP1B alpha is required for hepatocellular TPO generation, and you can tell from the left panel, we actually use the GP1B alpha knockout mice, and you can see in both serum and plasma level of TPO decreased significantly compared to the wild-type littermate as well as the beta-3 knockout mice strain. And also the liver TPO MRI level and also the liver TPO protein level mirror the results here, and it shows is indeed like decrease the TPO generation of the hepatocytes. And also we further pinpoint, okay, GP1B alpha is critical for this process, but which part, whether it's the extracellular part of GP1B or cytoplasmic part. So we actually further use the L4 receptor GP1B alpha transgenic mice, which we replace the extracellular domain of the GP1B alpha with L4 receptor, but remain the other complex of the GP1B, and we observe the similar results. We see decreased serum plasma TPO and also decreased MRI, TPO MRI and protein level from the liver. Based on her research, but there's still something we actually quite don't know. For example, like how does these GP1B alpha receptor on platelet surface can actually induce the TPO generation of the hepatocytes, since we have the sinusoidal endothelial barrier existed. So then we came up with the idea about what the role played by the Kupffer cells in this situation, because we know that Kupffer cells are liver residential macrophages, and it has the can contact with both platelets and also have close contact to the endothelial cells, and also they have interactions with hepatocytes. And also now it's well known that the Kupffer cells play also a very important role for the clearance of the decylated platelets or the aged platelets in liver. So the first thing we try to look at the function of the Kupffer cells is, as usual, we need to deplete it to evaluate the effect. So we actually transfuse both normal platelets or decellulated platelets, which they lose the salicylic acid of the platelet surface, and we transfuse those platelets into either control mice or Kupffer cell depleted mice by cryogenate. And as you can see here, for both plasma level and serum level, you can see the induction of the TPO generation in the control group mice, and also in both conditions, in both plasma and serum. However, the mice who lack of Kupffer cells, they failed to induce the TPO generation. You can tell the peak is around day 3, and then finally is also back to the normal level at the day 7. And we know that cryogenate depletion deplete both monocyte and macrophages, not only in liver, but also in spleen. So to further confirm it is a Kupffer cell's function, we also use a conditional knockout mice to specifically deplete Kupffer cells in liver. And we confirm the similar result here, but for the time I didn't show here, but we also do have the confirmation from our conditional knockout mice model. And we know that since TPO, one part is regulated by, can be internalized by the platelets, so in the condition of thrombocytopenia, you have less platelet count, so you expect to see a higher level of TPO in circulation. It is true in some disease condition. However, in the immunocytopenia, we actually observe like lower TPO level than we expected. So, and also I have to say that in a lot of ITP patients, we also have the autoantibody target to the GP1B. So we want to ask whether it also play a role in TPO generation. So we actually put the monoclonal antibody NET-E and to give into either control mice or the Kupffer cell depleted mice. And then when we use these monoclonal antibody, you can see they failed to induce the TPO change in vivo. And we know that is the antibody targeting GP1B. So which part? So we try to map the epitope. So thanks for we have a collaboration with Dr. Lupus lab, like Seattle group. So we actually did the test that these NET-E also block the GP1B alpha von Willebrand factor A1 interaction. So now we further pinpoint, okay, it is the VWFA1 domain that has played a very important role. And also with the collaboration, we also confirmed that this NET-E also can block the thrombus growth in the perfusion chamber. So the next thing we try to look at, even we think the endothelial cells is kind of like a physical barrier, but it's definitely not solid. We have like, you know, kind of like a windows for this endothelial cells. So that's kind of like little space, the pore, enable the hepatocytes, they have the micro protrusion, which can actually probably can get contact with the platelets. So, but however, we didn't, before the study, didn't carefully look at that part. So the first thing we do is we actually use the arsenite to treat the mice. And as you can see from here, so in the control, you have the fenestra, like in the endothelial cells, and however, after the treatment, you can tell like they have like a decreased fenestra area, and also we test the TPO level for this two group, and you can clearly see that once you have this pore disappeared, you have lower level of the TPO generation. And also next, we want to look at whether Kupffer cells also play a role kind of like maintaining this fenestra area. So the answer is yes. If you can see from here, so in the control conditions, we have the you can still see the nice structure of the little holes, the fenestra area, as the arrow indicated here. And however, when you deplete the Kupffer cells in the mice, and you can tell is actually, you cannot see a lot of these small windows in the endothelial cells. So we also quantify it. We have like, you have less porosity, and also you have less number of these little pores, and also we measure the plasma TPO level also lowered in the Kupffer cell depleted mice. And however, we didn't see any difference regarding to the platelets numbers in these mice. So since Kupffer cells play an important role to maintain this or promote the hepatocyte protrusion access to the platelets, so then we further look at how these three are interacted together. So you can see from the confocal images, we have different antibodies labeling both endothelial cells, Kupffer cells, and platelets. So you can tell from here, this is show the percentage of the platelets co-localized with either endothelial cells, Kupffer cells alone, or co-localized with endothelials and Kupffer cells together. And you can tell, so with the help of the Kupffer cells, you have more platelets can adhere to this area. And once you have Kupffer cell depleted, and you have definitely less numbers of platelets adhere to the endothelial cells. And through this transmission electron microscopy images, you can see from here, you have the Kupffer cells colored with blue, and also platelets in the red, and the hepatocytes in the gray area. And you can actually see from here, you have like the protrusion from the hepatocytes, which can close contact to the platelets. And also you have the Kupffer cells here. So we think about is before the Kupffer cells take up or engulf the platelets, is actually can kind of like hold this platelets and help them to better get contact with the hepatocytes. And also we have the quantification here. So because of the time limitation, so I cannot show all your data from this paper. So my poster is number 5, and there's a QR code, like you can scan it and get access to our paper. And I really, so here's the conclusion. So hopefully the take-home message here is, first is the Kupffer cell depletion, abolish the platelet-mediated circulating TPO, and both platelets, GP1B alpha, and Kupffer cells are interdependently required for TPO upregulations. And the extracellular domain of the GP1B alpha, which specifically BWF1 domain binding area, is responsible for the TPO generation. And also, Kupffer cell promote hepatocyte sinusoidal protrusion and facilitate hepatocyte-platelet interaction, which can trigger the TPO generation. So this study provides new insight into the regulatory mechanism of the TPO, and is also relevant to the liver function, which we can look at in the future study. So here I really want to acknowledge first my supervisor, Yu, like he actually bring me from the area of immunology and then to the platelet biology research area. And especially for this paper, I really need to highlight Danielle Caracas. She is a previous our PhD student in the lab, and the majority of the work and also the concepts are from Danielle and Jun Li, also another previous PhD student. I feel great honored I can join the lab, even though for this project I joined very late, but I learned a lot from this process, and also I also contribute the experiments and also to complete this work. So also I would like to thank our collaborators and also especially Dr. Lopez here and all the funding agencies. So thank you. Any questions?
I should know the answer to this, but if you infuse glycocalycin into the mice, do they make more TPO? That's the extracellular domain of GP1B alpha.
No.
They don't?
No.
No. So we try it. We try pretty hard. In vitro, in vivo, so we put soluble GPMB alpha there, that's not working. You have to immobilize GPMB alpha to the beads. Then let the interaction with hepatocytes, that's start working. This is similar to antibody against CD4, if CD3. If you immobilize them, able to trigger T cell, but the soluble is not sufficient.
Maybe that's why those Paxin-2 deficient mice have thrombocytopenia, because you don't have the GP1B in the right position.
Yeah, very nice work. I wonder if platelet binding to VWF, or I mean, VWF is involved or required for this process.
Oh, yeah.
And the Kupffer cell, actually, sinusoidal endothelial cells have a lot of VWF, but Kupffer cell doesn't contain any, but contribute to more of a clearance of VWF. I wonder, VWF has any role in TPO generation? Why GP1B binding site specifically for VWF is involved in this process?
For the knowledge, I actually came across a paper. I think they use the also the mice model. They mark the Kupffer cells with they have like a select 4F to pinpoint the Kupffer cells in the liver, and they find the co-localization of the VWF with Kupffer cells, even though they are not produced, probably Kupffer cell not produce VWF, but they have like VWF on the surface of the Kupffer cells. I think it is a play a role like for interacting with the GP1B alpha, but also it's maybe not like it's critical, but it's like I think other ligand receptor like on the macrophage surface also contribute to this process, like the MAC1, like other like integrin pairs may also contribute to this process.
Thank you.
So direct answer for this, we also check von Willebrand factor deficient animal. That does not affect TPO in the blood circulation.
Any additional questions? Hearing none, Grace, thanks very much.
Thank you.